Welcome, everyone. This is Joanne Geer. I'm the Executive Director of the Biopharma Research Council, and welcome to our virtual annual meeting. We'll wait just about 1 minute to let a few more people log in, but I see almost everyone's here, so we'll start very shortly. Hi, I'm Joanne Geer. I'm the Executive Director. I've been just about 2 years here at the Biopharma Research Council. And I want to welcome everyone. Last year we were able to orchestrate an actual physical meeting, but our community has become so international that we thought this year we would take the virtual route so everyone could participate. But it doesn't mean we're not going to have some parties in the future. In brief, we are an association for scientists all across the biopharma research community. So that includes industry, academia, nonprofit, government, supplier labs, and the people who love them. One of our founders, Ronnie Schreiber, is now the president of our board of directors. And I'm going to introduce her to you so that she can say a few words in a moment. Ronnie founded Planet Connect with Rich Brandwine in 1995 and is now the company president. She's also co-founder and member of the board of the BRC. Within Lucent's Bell Labs Global Laboratory Network, she provided assessments for the government of Manitoba and various universities, Walt Disney, and other Fortune 500 companies. She also worked at Johnson & Johnson. Ronnie's career began in the medical pharmaceutical industry in a variety of research and academic positions focusing on biochemistry and immunology, where she developed immunoassays for oncology and cardiovascular therapeutic areas. She held positions at the Dana-Farber Cancer Institute, Arthur D. Little, Rutgers Medical School, and Orthodiagnostics, now Ortho Clinical Diagnostics. She has co-authored and published several papers in the area of immunoassays. Ronnie has a master's degree from Rutgers University School of Library Information Studies and a Bachelor of Arts in Bacteriology from Douglas College of Rutgers University. Ronnie.
Thanks, Joanne, and good morning, everyone. Planet Connect has been creating internal customized symposium trade shows for one pharma at a time. But after 20 years in the industry, we've seen many changes that suggested a need to create multi-pharma events that are local or regional. So travel was getting more difficult for many, time away from the lab was becoming more limited. And in addition, many scientists and technologists were being limited in the number of educational events that they could attend in a year. So we found a way to provide education and networking opportunities to a broad technical audience, many of whom are in transition. And we're able to do this through the creation of the nonprofit, the Biopharma Research Council. So the BRC has enabled cross-pollination of ideas and collaboration across many organization types. Some of the BRC's unique strengths have developed through its association with Planet Connect, which has deep industry connections and expertise in event planning. Now, 6 years later, the BRC is thriving, and we hope it'll answer many of the educational needs of its target audience. So thank you for listening to me, and now I'd like to introduce Tom Fair. Tom is the BRC's Director of Strategic Alliances. He was at Merck for more than 13 years, where his roles included lead for the Nucleic Acid Capabilities Delivery Program and Director for Technology Licensing Integration. Prior to Merck, he was Technology Leader at Sarnoff, Manager of Biosensor Development at Omicron, and Principal Investigator at the Naval Research Laboratory. Tom?
So what I'd like to do is say a few words on behalf of the BRC and the great work that's been done over the past year. I think what we've learned from the webinars have been the great breadth and depth of work that's going on out in the community, and the BRC's attempt to bring that information. It's ranged from medical devices to exosomes to a whole host of other research areas, including topical issues like Ebola. So I think the value of the BRC in meetings is that ability to go out and find new and cutting-edge work and to bring it to your desktop, to be able to engage in those researchers who are doing the cutting-edge work and have them address your questions. one thing I think that I'd like to also ask is that those who, the members who are on the line, if you could also provide us with areas of interest you might have as we look forward to the new year and out years on what types of webinars you would like to see. And we will take your comments and see what we can do of finding the best presenters with the best topics in the areas. Some of the things we'll be looking at, I know this year, Joanne, without giving away too much of your thunder, include things like genome engineering and related issues. So again, I think it's been a great year, and I think BRC has shown the value in its ability to bring that topical information to you. Thank you.
Thank you, Tom. We so appreciate your participation and your enthusiastic support. We always need that. So here we are. We are really, we're really about bringing people together in various ways with enough depth of content that it makes sense, that it might be interesting for you to come to a particular program, but that you're probably going to be exposed to information that you may not already have, or some content about what's going on across the field or another field that's related to yours. I'm going to talk about our programs, which tell the story much better than I can. We started this series called D2D, Data to Drugs and Diagnostics, this year. The pictures on the bottom are from an event that we did not do in the water. This was last June, and we did a 2-day conference. We had 500 people registered. We had folks from across the biopharma community and really some fantastic interaction. The gentleman on the upper right was the postcard we used and the marketing we used for a meetup session that we did afterward in August. So we now have a meetup group that's also called D2D, Data to Drugs and Diagnostics, that and the whole purpose of this concept is to build the cohort of researchers who are literate enough about what IT people can do for them so that they can actually meet in the middle, and so that IT professionals who are understanding more and more about what researchers need can find, and I don't think the term is actually common language. I think it's learning about each other's language and finding ways to work together. This particular program was sponsored by a wide range of organizations, and we do a stand-up survey when we go to conferences, and we ask different questions each time. And this time we just asked, on a continuum from being basically a straightforward IT person to being a straightforward all science person, where are you? And these were sort of the 3 big categories. And so it said to us that this large community of people who are equally in science and IT is a very important cohort to build. And so you'll see a number of those kinds of programs ongoing. Let's see. This is a great program. This is the 3rd time this year. We went to Durham, North Carolina, and this is kind of an omnibus, regional, show everybody what you're working on kind of program. I'm sharing with you our committee members. This is the first time we've worked with a committee on the ground, and it was fantastic. We have booked our date for next year, which happens to be October 29th, and we expect to see many of the same people. These groups did a really particularly wonderful work with both helping to build the program and spreading the word, inviting people as well. And yes, that is an invitation. If you see things you want to get involved with, we'd love to hear from you. I think I appreciate Tom noting our webinar series. I'm going to kind of fly through these, but just to give you a sense of the range of programs we're able to do through the webinar platform. So before and after the BRC program, we continued on with some great topics related to the integration of the IT world and research. Now, this is where we first met Dr. Kuo. He got involved with us to put together this really wonderful short course in the exosome, essentially, with 4 sessions last September, October. And as you can see, we can really draw from people all over the world to bring great knowledge. And virtually everything I'm going to show you is available virtually through our website. We have an occasional series we call the Mobile Patient, which looks at the way that the emerging network technologies are affecting what we learn about patients, what we need to think about, what are the regulatory issues, what are the security issues, and we have some really nice ones in the works for this year as well. In December, we did, at the end of the year, we did kind of a highlights of different ideas that seemed to be emerging, and we started with the introduction to the microbiome. And this is leading to, I'll share with you later, we're going to be doing a whole series this year on the microbiome. This was a particularly really intriguing program. And then these two are series that have started that are continuing this year. Flexible biomanufacturing is really lovely. We in January we did a program we called Data to Decisions. So it really looked at what data is being collected throughout the biomanufacturing process and how is it addressed and how does it assist in design and managing change and all that kind of good stuff. And the global collaboration to address Ebola is also an ongoing series. In April, we have Pardis Sabeti is going to give us a webinar on her work, and she's been profiled in the New Yorker and New York Times, and particularly wonderful researcher we're going to learn more about. We get this all done with a lot of help. We're a very small staff, and I just want to share with you the folks who are on our scientific advisory board. They stimulate ideas, they help us with outreach, and sort of carry around our story, which is an important thing. And this is a new group I'm very proud to announce. We decided that the Scientific Advisory Board kind of has its role, and as you could see, those are quite senior people. But there's a real need for an opportunity for early career scientists to play a role and to obviously help us, but also to have opportunities to do presentations and to build their skills in leadership through our involvement with us. We start with Devin Lambert, who actually was an intern with me some years ago at another role that I had, and who is now at Cold Spring Harbor PhD program. Danielle Macario has helped us out with, she's presented at one of the mobile patient webinars and is continuing to help with that. Satvik Gadamseti spoke in the manufacturing webinar in January, and Jeff Harris is just getting started, but we're already in the works on developing our meetup program that he's involved with. And so I'm sharing all these faces. Almost every one of these people just stepped up and said, "Can I help you?" And I said yes. And so we found a way to bring them in, and each one of them is really contributing a great deal. So I'm going to share our awards now. On the left is Kristen Ward, and those are all the awards before they got shipped out, and on the right is Danielle Macario receiving hers. But I have to say a few words about Kristen. She makes us look good. She is our art director, and we could not do what we do without her. The work that she does is great support, and her creativity and poise under pressure is really spectacular. So big thank you to Kristen. And now I'm just going to tell you very briefly about what some of these folks have done for us. Howard Young is at the NIH. He's on our scientific advisory board. He's working with us on the microbiome series. He made connections for us for the Ebola program. And just a general, he's one of those people I have to talk to all the time that's making wonderful things happen. Winston Kuo, you're going to learn a lot more about in a moment. Essentially, he's on our board. He worked on the exosome program and the microbiome series, and we have a number of things we're cooking up for the coming year. Rodolphe Barangu from NC State University is one of the pioneers of the CRISPR method of genome engineering, and he was a presenter and on the committee for our the Triangle Biotech Research Symposium in Durham, so we want to acknowledge him. Tom Johnson of ExoStar has helped us in a number of ways. He participated in the D2D program. He spoke at in our Collaboration in the Cloud webinar. And as a supplier, their company is all about federated ID on the web, so that programs that use their technology or companies that use their technology can allow individuals to log in once and then have access to all the appropriate tools that their company has purchased. So it's a really smart approach to working in the cloud. We think that's very important. Robert Bolin was our keynote at D2D and has been a generally great supporter. Francis Ligler was also a presenter at our Triangle Biotech Research Symposium and really led the way in reaching out to a number of our speakers and program development folks. You've already heard a little bit about Satvik Gadamseti and Daniel Macario, and we just want to acknowledge their help. It's really important to us to have folks at all levels of their careers. So I would like to give a virtual round of applause for these wonderful people, and there are many more. We have many volunteers. I'm going to mention someone. There is another group that's forming this year, which is a business roundtable, and I'm just going to give a shout out to Robert Hutchins from Witham Smith & Brown, who's leading the way for that. We have a number of partner organizations, and all of them help us with spreading the word amongst their communities and also with outreach, program development. This is a really important part of our growth. And this is just a small selection of our supporters over the last year. It's really been quite a year. So I'm just great thanks to all of the suppliers that have made our programs possible, both financially and content-wise. We really like the content that we're able to bring in from all these partners. The supplier role is really key in today's biomedical research world. There's a lot of problem solvers out there that are making things happen. And this is to give a special thank you to Planet Connect. I think Rana gave you a really nice introduction to what they're all about. And the part that she didn't underscore is that the meetings that Planet Connect organizes are proprietary. They're secrets. They can't go out and tell you everything that they've done for these companies. And there's been dozens and dozens of meetings over the 20 years, thousands and thousands of people brought together. And I really want to acknowledge the vision, the strategic support, the financial support, and the general wonderful attitude that Planet Connect displays in everything that we do. None of this would be possible without them. I should also say that when we do our programs, we rely on Planet Connect staff like John Honorato, who is our tech guru today and every day, and is another person we could not live without. There are also technical staff, there's support staff, sales staff, all of them come together to help us out with everything we do. And now we turn to our keynote. I'm very excited to introduce you to Dr. Winston Kuo. He has a really wonderful background, and as you could see throughout, he's been getting involved with us in a number of ways, and it's been a great joy. Winston is the founder of Harvard Medical School's Laboratory for Innovative Translational Technologies, which was an integral part of the Clinical and Translational Science Award program. He initiated and participated with the FDA in a large-scale comprehensive study evaluating the clinical utility of gene expression technologies, which was published in Nature Biotechnology in 2006. He sits on the NIH SBIR Grant Review Studies section and is a peer reviewer for the Qatar National Research Fund. His clinical and translational initiatives have expanded globally in developing countries such as Brazil, China, Mongolia, Nigeria, and so on, focused on accelerating understanding of mechanisms that affect human disease, catalyzing the identification and development of useful biomarkers, and speeding the development of therapeutics in patients. Dr. Kuo has also instructed in the Innovation for Economic Development program at the Harvard Kennedy School of Government. He's a founder and trustee of the Otto Weinrich Warburg Cancer Research Foundation. My favorite is that this summer he will participate in an Arctic expedition to sequence the narwhal with principal investigator Martin Nguyen of the Broad Institute. Winston sits on the editorial boards of multiple peer-reviewed journals, is the editor-in-chief of the Journal of Circulating Biomarkers and Nanobiomedicine, both of which he founded. He has consulted with the NIH on interdisciplinary outreach and sits on a number of business, nonprofits, and nonprofit scientific advisory boards, including ours. He received his Doctor of Medical Sciences, Oral and Computational Biology from Harvard Medical School. I turn it over to you, Winston.
Thanks, Joanne, for your opening remarks and kind introduction. Well, good morning, everyone. Good afternoon or good evening, and Gung Hei Fat Choi, everyone, too. It's Happy Chinese New Year's, the Year of the Sheep, and I really wish everyone good health and success in the upcoming year. I'm really privileged in speaking today, and I'm honored and grateful to give the keynote to the Biopharma Research Council community, the first virtual annual meeting. I have had the pleasure to engage, as you heard, with the BRC, and it really represents a good platform for increasing collaborative and disciplinary nature of current and future biomedical research. I'm currently the CEO of IES Diagnostics, but before I get into the details of what IES Diagnostics is and what my role is, I would like to share my experience prior to this industry position and then some concluding remarks. I guess during my graduate years at Harvard, I was sort of strategic planning my career. I had an excellent 15 years there as a grad student, then a postdoc, as Joanne had mentioned, comparing standardized different gene expression platforms, studying Darwin's finches from Galapagos, to breeding mice in the mouse room, to understanding the roll the tusk of the narwhal from the Baffin Islands. As a grad student, I transitioned to an instructor, then to assistant professor, and I knew I needed to do something because I knew I was getting older, and I know I didn't want to stay in the academic track for a long period of time. So whether I go to practice dentistry, as I'm a dentist, or go into industry, but I wasn't sure at that moment. But I knew I had better learn how to reduce cycle time and strategically position myself, hence turning discussion into tangibles. So after some vetting, this process I'm going through, I thought I had an opportunity. After my paper was published in Nature, I knew I had to plant seeds prior to that. So as I mentioned earlier, for my next challenge, which I didn't know at that time, however, I needed to do something impactful, I would say. And as you can imagine, to start up anything at Harvard is always daunting. There's so many hoops to jump through. There's logistic issues, costs, what are the returns to the medical school, the benefits, the risks, and etc. But I was up for the challenge. I started drafting my business plan to create an entity, that sort of can, benefit the Harvard community at large. My laboratory was launched in 2007. It was called the Laboratory for Innovative Translational Technologies, as Joan had mentioned, and it was originally created and located at the dental school, and I was trying to open it up to the medical school, so to have early access to enabling cutting edge genomic and proteomic technologies. The whole idea of this model was to sort of bring together a myriad of technologies in one place and put them in the use of the hands of investors and thought leaders, and which would bring out fruitful discussions and would bring things that are sort of not mainstream, right? This is out of the box type of thinking. So many researchers with highly focused research interest projects are definitely not aware of these nascent technologies that emerge from industry or academics. So this is the future of Complexion, a biomedical research organization giving the pressing need for integrated interdisciplinary approaches to solve challenging clinical problems. So I was committed to breaking down these barriers and to increase the awareness of these novel technologies among local and national researchers. I thought this collaborative model can accelerate the process by generating high quality data utilizing these accessible technologies. So as Joanne alluded, so predicating CARVCAS, the Clinical Translational Science Center, which is funded by NCATS, which is the National Center for Advancing Translational Sciences in the time of precision medicine by a year, but premise in its mission, the lab was designed to accelerate laboratory research in the therapeutic and diagnostics into the clinic. So in doing so, there's many tangibles that came out of this. It established a huge network of industry, academic collaborators at the local, regional, national, and international levels. We also established a regulatory and administrative infrastructure, right, that's necessary to create and manage complex networks involving multiple academic and industry efforts. So in an effort to reach beyond traditional research and deliver paradigm-changing outcomes, my group had created a community of healthcare providers by implementing the Identify, Sustain, and Enable methodology that sort of promotes and sustains communication and collaboration amongst this community of scientists, engineers, clinicians across the many Harvard Catalyst institutions via sort of a clinical pull and not the technology push approach. So my lab combined an expert-based approach with active, continuous grassroots outreach to identify and assess, clinical unmet needs and the relevant stakeholders that sort of can solve these problems. So it was exciting in creating the self-sustaining community to solve these needs. So the projects included microbiome, looking at the oral, gut, the fecal area, DNA repair, radiation oncology, sepsis, looking at shock and post-cardiac arrest, circulating tumor cells and microvesicles, from biomarker discovery to validation in the areas of oncology, neurodegeneration, blood storage, autoimmune disease, infectious diseases. It's endless, right? So the community and network became a fertile ground for the formation of also public-private partnerships. And the tangible of our network model generated enough broad-based interest, for example, in the field of exosome and microvesicles, to play a significant part in launching of the Journal for Exosomes and Microvesicles within tech publishers, now which is called the Journal of Circulating Biomarkers. So the ecosystem, the Harvard ecosystem, has really given me a foundation to work with. So I had the ability to gather scientists, engineers together to formulate an expert-based approach, and I learned to understand people, right, able to put together functional teams that can sort of enable individuals and yet deliver a tangible at any given period of time. Every day to me is I do an experiment, right? Whether it's something that I do in the lab, whether I'm sectioning, whether I'm conducting laboratory experiments, or even pushing the envelope with Harvard administration, actually was able to test everything and on all fronts. So more often than not, people, on boundary crossing expedition discover something they're not looking for, which is either serendipitous nor careless investment of their time and energy. So exploring something new in unfamiliar topics or places with different types and kinds of people can provide seeds for collective intelligence that, not all boundaries are geographical. So let me plunge a little bit and talk a little bit about IES Diagnostics and how I got into the company and what my role is. So IES Diagnostics is a molecular diagnostic company that has a proprietary patent-protected interferon-based assay test. From here, I'll just call it an assay that was developed at the FDA. And we licensed, we have a license that's exclusive to our company through the NIH. This new assay called ReactImmune is an interferon-based assay that's capable of detecting complete interferon expression signatures for many diseases, so which can be utilized in diagnostics and can also be used as a tool for monitoring treatment of a variety of related diseases and illnesses. So the interferons, just for those to keep everyone on the same level, is a family of cytokines. They constitute a first line of innate defense against viral infections, more specifically type 1 interferons, including interferon alpha and beta, activating toll-like receptors, which are a class of proteins that play a key role in the innate immune system, and non-toll-like receptors in the cascades. The company is founded by our scientist, Dr. Ron Jubin, who has over 2 decades of experience in the fields of virology and immunology. We collaborated on both lupus and ovarian cancer interferon profiling projects when I was at Harvard Medical School with the folks at BIDMC, Beth Israel, and also the Brigham Women's Hospital. But the funny thing is that he was introduced to me by my classmate, Steve Greenberg, who is now the director of neurology at the Brigham. And we were students back at MIT in 2001. So you can envision people, relationships, networks are good. But, one of the reasons why I probably got the job was because our tangibles, right? we were able to generate data in a short period of time with lupus and ovarian patient samples, and we saw unique profiling based on interferons. And this is sort of how I got myself into this job and looking for an exciting opportunity. So we're a great team of less than 10, very lean and very efficient. So now let me get into a little bit about our technology. What makes it so innovative, and why is it important? So our body's immune system produces proteins, as I mentioned, are called interferons, and then what they do is they combat illnesses and diseases. So interferon-associated diseases that include autoimmune disease like lupus, rheumatoids, multiple sclerosis, as well as cancers and viral infections. And in these cases, our immune system is dysfunctional when turning the interferons on or off. So in time of personalized medicine, a key aspect to treating these diseases is to identify a therapy that's sort of capable of properly regulating these interferons to return our body into sort of a healthy state. So each disease triggers our immune system to engage a distinctive combination of different types of interferons. So you can imagine when we're profiling lupus or rheumatoid arthritis, they would each one have a different interferon profile, like a fingerprint. So this is what we call our interferon expression signature, okay, for that particular disease. However, on the downside, without the signature, diagnosing interferon-related illnesses will remain really challenging, and more importantly, can't determine the best course of treatment for a particular type of interferon-related disease. So as of today, I mean, to the best of our knowledge, there's not an assay that sort of is available to determine this type of signature. So currently the course of treatment is to hit these infected cells with a broad range of immunosuppressants with the hopes that some combination of the interferons contain. But there are a lot of risks associated with this, right? So using unnecessary immunosuppressants weakens the body's immune response and making them more vulnerable to all other types of ailments that can often come because of that. So why is this important? So our assay, the reactive immune assay, so far can accurately and reliably determine the signature that's specific for that particular disease with that profile. In addition, the assay will dramatically, which is very important, as we're sort of partnering with pharmaceutical companies, shorten the drug discovery process for the interferon-based therapies. You know, it will save these companies billions of dollars, both in time, money, and provide those affected with this, the specific disease, the highest clinical benefit with the fewest side effect at low cost. If you saw on the page, in reference to the webinar, and we actually recently took center stage during the midst of the Ebola outbreak. When it appeared initially, Ebola was sort of contained in West Africa. It was becoming an increasing concern and more challenging. So as of last week, I just looked at the CDC, the World Health Organization, reported there's been 23,000 cases and almost 10,000 deaths in 9 affected countries. So we had questions as it was related to how our technology can address some of these concerns last fall. But given the recent uptake, Ebola virus is sort of somewhat contained, and there are decreasing cases, like for example in Liberia and also the regions. But the questions like how far will it spread, and at what rate? Can a diagnostic test be developed to determine whether a patient is infected or exposed, but presents as asymptomatic in order to shorten or avoid quarantine? As epidemics continue to come and go, can we develop testing to differentiate between bacterial and viral infections, right? Because the symptoms are very similar. And as the global community embraces Ebola virus epidemic, how do we move forward in the absence of effective early diagnostic tests and vaccines or therapeutics? Or can there be a companion diagnostic test to be able to monitor the effectiveness of a vaccine or a drug that's already in development? So why interferons in Ebola virus infection, right? So unlike other viral infections, Ebola has evolved to evade detection by infecting dendritic cells. These are your antigen-presenting cells. Inhibits signaling of cytokines to eliminate the virus. And there are a couple Ebola viral proteins, and thus allowing the progeny of these viral particles to invade the cells unchallenged, causing the infected cell to die and to hemorrhage. And these are the characteristics that you'll see. So dendritic cells are specialized cells that sort of play a central role in our innate and adaptive immune response. Upon an infection, dendritic cells have the ability to uptake these particles or microbes by phagocytosis, process and present them to other specialized immune cells like CD4 T cells that play a sort of cardinal feature in adaptive immune response. So dendritic cells sort of initiate this adaptive immune response by activating the CD4 T cells, and more importantly, these APCs can guide the immune response by inducing CD4 T cells in a particular cytokine release pattern that will result in a specific immune response that targets these pathogens. So interferons are among the tools utilized by these dendritic cells to sort of regulate the adaptive immune response in CD4 T cells. And the immunopathogenic properties underlying this Ebola virus, it really is multifactorial and very complex. So the benefit of our assay, as many pharmaceutical companies are in the process of developing targeted therapies against these viral proteins, where inhibiting the target will result in increased production of interferons. So this specific interferon family is a potential prognostic biomarker for survival for patients infected with Ebola. In addition, we can also, and we're in the process of co-developing a diagnostic test with Ebola virus signatures and interferons from our assays that will enable clinicians to monitor and measure the effectiveness of these experimental therapies until approved by the FDA. And this can be utilized for screening, and where the interferon levels with patients affected would tend to be low, complementing existing Ebola tests as compared to other viral infections like influenza and malaria. So what are some of the lessons we learned from this outbreak? This is a good segue. We must prepare ourselves now in the future, not just only for this outbreak, for many other things, and as well as learn from prior experience, including SARS, H1N1, H5N1, and the most recent one, the Middle East respiratory coronaviruses. So these are samplings of a few things that have happened, but we can be more anticipated epidemics and outbreaks of new and or re-emerging infections. So we feel that interferons play a really critical role, not only in our immune response, but mechanisms that interfere with the production of interferons, also potential targets for therapeutic development. So my role as Chief Operating Officer is really to drive the commercial assay. By fine-tuning the assay for those particular diseases that are in our interest right now is lupus. We are working with collaborators on Ebola virus right now, implementing our diagnostic pipeline, clinical diagnostic pipeline. We also are, my role is to seek key strategic partnerships that are local, national, international, both academic-based and industry-based for our proprietary and partner assays in the future. In addition, we will work closely with the FDA, and we will eventually apply either for LDT or 510 approval process. And then lastly, the most important part is to sort of establish a reimbursement strategy for our assay. So in the end, people I've met, I've been really grateful for the people I've met. And, and the decisions I've made throughout my career has made me where I am today. I've been very fortunate meeting great people through my journey, although at times, life appears very daunting. I think, not everyone has the luxury to be within the hard environment. It's really harsh out there. And without people, right? I could have great technologies, but without people, none of these things would matter, right? I learned to communicate across borders, develop relationships, collaborations, and how to manage those relationships to turn our discussion into something tangible. In academics, what's tangible? A grant, a paper, you have press releases, you give talks, you educate. In industry is totally different. You do the same thing, but the main priority is to make impact to our patients. So we are lucky, and I think kids are lucky nowadays. Because they get to leapfrog, in reference to technology, right? My son didn't know what a landline was, right? Because he grew up with the cell phone, and right now with social media, this is a new paradigm shift in how we communicate today. I hardly use this, but this is the way that of the future. So giving communication is always at your fingertips, and learning how to best leverage this capacity without losing sight that face-to-face communication, I think, is still the best way to engage and get things accomplished. But another important aspect I realize is that we meet people for a reason. The reason might not be apparent immediately. Maybe this afternoon, maybe 10 days, maybe a month, 5, 10, 20 years from now, you don't know how these relationships or the relationships of these people that you interact with, how they can come back in and out of your life. So don't burn bridges and be diplomatic in your interactions. And also, the most important thing I realize is that when you engage with people, be sure you have something solid that sort of comes out of this, out of your discussion, because you don't want to waste people's time. I don't want to waste my time, and also don't do things that you regret. So, as I said before, being in the industry is very different than an academic environment. I continue to engage with the global life science community, but based on my prior experiences and my network, so one example how I can connect with folks pertaining to my interests was to create a journal, creating a global community via journal, just like what the BRC is doing. And as I mentioned, during my last year at Harvard, I was able to co-found a journal called Journals Circling Biomarkers, which encompasses hot topics like exosomes, circulating tumor cells, cell-free DNA, and more recently, last year, we created another journal called Nanobiomedicine that sort of covers the cross-disciplinary area of intersecting nanotechnology with biomedicine, and both with InTech publishers. This is a great opportunity for me to sort of engage with faculty, reach out to folks from industry, understand and see what students are doing globally, how they contribute, for me to read about the hottest research topics. In addition, just to say, because of my relationship with both the BRC and InTech publishers, we're able also to create a partnership between the two entities to co-host future webinar series with related and associated publications. Now for some food for thought. Knowing how challenges in academia and in the marketplace, there are many opportunities, but sometimes they're hidden. I always tell my colleagues, don't wait for the opportunity, because they'll pass you by. Figure out how to sort of ignite yourself. Identify, leverage the strengths and the weaknesses, and be proactive and apply yourself to current and future challenges. It doesn't have to be the healthcare sector. It could be anything, right? Because the things that we learned in here can be applied to another area. So interact with people in the ecosystem you want to engage in. Figure out why and how you can contribute. What is the tangible people are looking for? And perhaps maybe they haven't thought of it yet. So as I mentioned, life is full of opportunities, and at times may feel bleak. And that time, try to create your own opportunity. So everything in life does not last forever. There's always a window of opportunity. And hindsight, of course, is 2020. Took advantage of my accomplishments, and I learned to take chances at Harvard. And the answer, if you don't take chances, will always be no, unless you try. And to me, there's no such thing as luck. Well, if you win the lottery, that's a little bit different. The innovation I created at Harvard Dental School was incorporated into a large initiative at the medical school a year later. So that luck that I had was my preparedness, the risk I took that met the opportunity that came at that time when NCAS decided to give these awards out nationally. So try to leverage your experience and get engaged at local, national, eventually internationally. And this is a good way to sort of brand yourself and make good associations with the people, the group, nonprofit, for-profit companies to be able to accelerate, reduce the cycle time for yourself. So I have realized, reflecting back on my life as of today, the things that have made impact to my career was my tenacity to do better, never give up, and keep navigating. I've had great mentors, I've met great people. I've learned to be more creative. I've learned to find balance within myself. And everyone is different. Engage with diverse cultures and always give back at some capacity to society or your community, just like what the BRC is doing. And at the end, you need to learn to believe in yourself. Building that confidence is important, but also at the end of the day, having a tangible will fly light years ahead for yourself as a career. So everyone's journey in life will be different with ups and downs, and it's inevitable. I learned not to live life as I should have, could have, would have. Live smart, because life is dynamic, and behooves you not to live life to the fullest with no regrets, since life is too short not to be happy and unaccomplished. Thank you. Gung Hei Fak Choi. Again, I want to thank Joanne, the Biopharma Research Council team, for the invitation, allowing me to engage and contribute to the BRC, and I look forward to an exciting year. Thank you. Joanne?
Thank you so much, Winston. That was really fantastic. I would like to make another virtual applause here. And I learned a lot about you today. I will say that absolutely Winston lives exactly what he's talking about, and I like being in the position of saying, wait, we need to figure this out. We need to figure this out, because Winston is always challenging me with new concepts and thoughts about what we can be doing. So thank you so much.
My pleasure.
I see some really nice comments that I'm going to share with you, and if anybody has questions you'd like to add, that's great. I have one from Sam Consummate, a couple from our scientific advisory board. Thanks, great talk, Winston, very inspiring. Happy New Year, and virtual applause to you. So
thank you.
And I can see one of the things that's fun about the hosting piece is that I can see that virtually everyone who has started with us has stayed with us through the whole time. I'm just going to take a moment and talk a little bit about what's coming up next, and then we will adjourn. But just thank you so much, Winston. This is a lot of fun.
It was my pleasure. It was my pleasure. Thank you again.
Thank you. So I'm just going to mention a couple of things coming up. There's a lot more, and all of it will, of course, be reaching you throughout the course of the year. But Winston and Howard Young, another member of our scientific advisory board, are working very closely with us to develop the short course in the microbiome, which is a webinar series in April, different topics throughout the first 4, and then the bonus round in May is going to actually be a roundtable. We'll have about 6 different experts talking about different aspects of what's going on in microbiome research and breakthroughs that are relevant to health. And then I'm very pleased, we are going to have a full-day conference at Fort Detrick in September. And lots more detail about that, but that will be free to attendees. So if you're in that area or can make it to that area, that'll be a really terrific program, probably about 200 people. Coming up in May in Boston, this is really thrilling. This is something that is being hosted by Merck Research Labs. This is Advancing Drug Discovery Through Genome Engineering. So we're looking at CRISPR and TALENs and zinc fingers and different models that are being addressed, and the registration is open on that, and I'm already seeing some great enthusiasm. So that's a one-day program, and very, very interesting stuff. It's also on our website already. And I think I mentioned before, we're going back to Durham for October 29th, and the committee is building for that. Many of the people that were on the committee last year are involved again, and if that intrigues you and you're in the Research Triangle Park area, we would love to include you in the planning. This is going to focus this year on technologies that go across human biology, animal biology, ag biology. So it's kind of a universal technology program this year. And thank you. I want to thank everyone from Planet Connect for making everything possible. I want to thank all of our boards and communities. I want to thank you for being part of it. We're going to be posting this recording on the website, and we'll also be sending out a paper version of our annual report, so you'll be able to take a look through. Maybe there are people you know that have been involved with us throughout the year, and I encourage you, you can reach me very easily through our website